Hello students, welcome to Turito live classes. Myself is Swagat. Today in this particular session, we are going to understand about blood grouping. So that is the topic for today. So let us begin the very same chapter, that is life process. So in this chapter, today's discussion will be completely on the blood grouping. So before we go into the topic, let us look at the topics what we have finished in the previous session. So in the previous session, we tried to understand by recollecting the components of circulatory system. So we try to recollect the different components of the circulatory system, starting with heart, blood vessels and blood. So these are the main blood components of the circulatory system. Next, we understood blood as a fluid connective tissue. So the nature of the blood itself is fluid, and that is actually a tissue to a category that is a connective tissue, right. So this is what we understood, and we saw there are different components of the blood, including erythrocytes, leukocytes and platelets, and we call platelets as thrombocytes. So we understood this in the previous session, and we also looked at the details of the formation and development of the blood cells. So can you tell me, children, where do we get to see the formation of the blood or blood cells? Okay, yes, some of you are putting in the chat box, I see that. Okay, now what we can understand is this formation of blood cells actually takes place mainly in the bone marrow. Bone marrow. Now in the bone marrow we have two types, right. So that is available in the bone, right. So it is the red bone marrow and yellow bone marrow. Now we have to understand that the complete formation and development of blood, that is hematopoiesis, is the process of formation of blood, right. It includes formation as well as development of the blood cells, okay. That takes place in the red bone marrow of the bone marrow, right. So that is what we understood in the previous session, and followed by that we try to classify the blood cells, that is we actually tried to classify the white blood cells. White blood cells were classified into two types, they are granulocytes and agranulocytes. So when it comes to granulocytes, we see neutrophils, basophils, eosinophils. When it comes to agranulocytes, we see there the presence of the lymphocytes and monocytes. So that was the classification of the WBCs we did, and that stands out to be granulocytes and agranulocytes. So this is what we did in the previous session, and then we also understood the blood as it has different components. So when you look at the blood components, we looked that blood is also having different components, plasma, which is mainly having that yellowish color, okay, following which we have various blood cells, they are of varied colors. But whenever we bleed out of an injury or any scratch or anything happens to him and we get to have a wound, if we bleed, we see that our blood would be having a red in color, which means we get to see the most abundant blood cells in our fluid connective tissue is RBCs, okay. That is the most abundant blood cells what we see, right. So RBCs are the most abundant blood cells in the body, following which we have the platelets. Platelets would be the second abundant blood components we get to see. After that we see it is the WBCs, they are the least abundant cells in the blood cell, right, among the blood cells. Now that is what we have seen, and we also understood the different details of erythrocytes, leukocytes and thrombocytes. So, about their abundance and what do they do, what kind of their size they are, which of them is having the largest size. So, when it comes to that, it is the leukocyte, that is the WBCs, they have the largest size among the erythrocytes, leukocytes and thrombocytes, okay. And they are also involved in immunity, that is acting against the diseases, that is what is performed by the leukocytes, okay. So, this is what we have done in the previous session. Now, let us have a look at the learning objectives, what we have planned for this particular session. So, in this particular session, we are going to understand about the various learning objectives, what we have planned. So, fine, let us see here. First one, we will understand how the discovery of blood grouping happened, okay, who discovered it, okay, that is what we will be looking into. Once done, we will also look into the discovery of the antigen from a rhesus monkey. So, presence of this antigen in the blood is one of the important thing, okay. So, that is entirely different thing. And we also going to understand about mechanism of blood transfusion, and we will understand about few details of universal donor. So, with respect to blood transfusion, we are going to understand about universal donor, and then we will also understand the distribution of blood groups in the population, okay. So, this objective will be pertaining to the Asian subcontinent only, okay. So, we will look into that details. After that, we will also understand the process of blood coagulation, or in other words, the very same blood coagulation is also known as, we are going to understand about blood clotting, how blood clots, what is that process, what is the mechanism, we are going to understand that in short, okay. Then we will see circulatory system and its types. So, in our body what kind of circulatory system we have, and we will also look into the types of circulatory system. So, there are differences depending on the type of the organism. So, we will look into the types of circulatory system. So, this is what we have planned for you today. So, let us begin with this blood grouping. So, before I start something about blood grouping, I want you students to put in the chat box whatever you are aware of this blood grouping, okay. So, let us have a few details mentioned here. So, you know blood grouping, and I see many of you are dropping in the types of blood groups what we have, okay. That is A, B, AB and O. These are the main groups what we have, okay. And we will also see that I see children are dropping in about A positive, A negative, okay. At the same time B positive, B negative, and again AB positive and AB negative. Same stands with O positive and O negative. So, this is what you know so far, and you might be knowing that it is the O group which is usually known as the universal donor, okay. That is what we know, right, okay. So, and you also know you know different blood groups, you know transfusion, blood transfusion, that is a person giving blood to another person who is in highly need of blood, that is what is, you know, giving the blood to another person is what is transfusion, blood transfusion, right. These are simple things, okay. And you are pretty much aware, and this presence of positive and negative thing, how it happens, what exactly is that. So, we are going to understand now, okay. So, fine. So, let us begin here. So, do you know, like, before we can go into these details, can you tell me who started this concept of blood grouping, this system of ABO blood grouping, who started that? Think of it. Okay, correct. So, I see many of you are dropping in the name Carl Landsteiner, okay, or Landsteiner, depends on the pronunciation, okay. So, Landsteiner, so he was the person, okay. So, let us see. So, it was started by this man, okay. We have to thank him in a way to understand about the blood. So, it looks different, sorry, it looks same for everyone, mean the color, but looking at it there are still differences. So, when you look here, this system of ABO was discovered by an Austrian scientist. He was an Austrian fellow, name is Karl Landsteiner, in 1900 AD. In 1900 AD he discovered it, okay. So, that is very much important, even in various other subjects also, and even in science we get to see who did this work, okay, and when it was done. So, the same thing we are also following here. He was the one who did this identification or discovery of the ABO system of blood grouping, and that was achieved in the year 1980. Okay. So, ABO blood grouping is based on the presence or absence of two antigens. So, you might wonder, when I am saying about the presence of the antigen, and in some of the previous discussions or in the previous grades, that is class 9th, you might have heard antibodies are present in the blood and they fight against the diseases. That is what you have heard, right. Even in previous discussion about WBCs, we might have that usage, antibodies, okay. But you see there are antigens also present in the body, that too in the blood. We normally consider antigens, we consider antigens usually as foreign substances, right. We normally consider like that, the foreign substances, isn't it? But what is the science here? So, we are going to understand that now. So, what exactly is that biology here? So, we are going to understand now. So, we get to see the presence of the antigens on the surface of the RBCs, and they are named as A and B, and we have antibodies in the plasma. Okay, that is what we have seen. Let us have some more understanding. So, it was this person who tirelessly worked to understand the system of blood grouping, ABO. It is after him we, even now till today, we understand blood groups with their usual possible combinations, presence of A positive, A negative, B positive, B negative, and we have AB again positive, negative, and we have O positive, negative. So, it was his work, right. So, we have to understand his work also, fine. So, this is one thing. What next I have for you? You see here donors and recipients. So, this is what I just want you to understand in brief. So, I will be telling you in short to make it a bit easy, you have to follow that, okay. So, here O group individuals are usually universal donors, O group individuals, okay. And then we see AB group individuals are usually universal recipients, they can get blood from any other groups, whereas O group individuals are universal donors, they can give blood to different individuals, okay. I want to show you a table, see here it shows few details here, okay. So, we are going to understand this table, it is of course having some of the limited information here, but then we are going to analyze the same concept in the upcoming slides in a different way. Fine, let us have a look here. So, when you observe this particular table, you would see it is all about the blood group A. Blood group A has antigen A, okay. So, it has antigen A within itself, and what about antibodies in the plasma? So, the person with blood group A plasma is different, we have learnt earlier, isn't it? And blood cells are different, we have learnt that earlier. So, when it comes to RBCs, in the RBCs, you know, we get to see the presence of the antigen. So, this antigen are actually on the surface of the RBCs. So, here in for the blood group A, antigen A is available, okay. Antibodies in plasma, that is anti-B is present. So, the type of antibody present in blood group A is having plasma. So, that plasma contains antibody B, and then you see who are the donors for this group. So, the person who is having blood group A can receive blood from A and O group. Okay. So, we will understand that differences also what we have. So, likewise you see here for blood group B, you see the person will be having antigen B on the surface of RBCs of the blood group, following which you will see antibody A will be present here and the donors are B and O. That is quite simple, right. So, again the person with blood group AB would be having both the antigens on their RBCs, that is A and the B. So, it is a common antigen, you saw that in A, you saw that in blood group B, and even now in blood group AB you see the same antigens A and B. So, let us move forward. You will not find any of the antibodies here, no antibodies in blood group A and B. So, let us understand that later. So, who can donate? AB, A, B and O. So, you see there blood group, the individuals with blood group AB are called universal recipients. So, who can donate? AB, A, B and O. Person with AB blood group, A blood group, B blood group and O blood group, they all can donate blood to the individual with the blood group AB, right. Now, you see blood group O, again you see you will not find any of the antigens here, no antigens in blood group O. When it comes to antibodies, they have blood group antibody A and antibody B in their plasma, okay. Antibodies are present in the plasma, okay. And who can donate blood to them? They can only receive blood from the O group individuals. Now, after going through this particular table, many of you might have a question, that is blood group A can receive blood from A and O, that is what you said, you might be asking that way, right. So, we have to understand, if it is A and the person has to receive blood from the very same group, then it has to be A positive should give to A positive, A negative has to give it to A negative, okay. But there is a problem here, I know many of you might be having problem, that is with O, again O here, O, O. So, what is the problem with O and AB is the individual having AB blood group, they do not have antibodies here in the plasma. The individuals with O blood group, they do not have antigens in their on their RBCs, okay. So, this is in short about the universal donors and recipients thing, okay. Now, please keep this in mind, okay. We will proceed further, you have to link some of the last column details with the upcoming details. So, let us see here. So, I was just telling you that AB positive or AB negative, A positive or A negative, we differentiate the blood groups within the same category, it is positive and negative. So, where did we get that categorization? That is what we are going to understand now, okay. Let us see now. So, another antigen, it is Rh antigen, okay. So, when it comes to this so there are so many antigens available, by the way, but for the first time, we saw the antigen, that is RH, like one present on the rhesus monkey. So, that presence of an antigen, okay, you see here antigens. So, this is what it is. So, we normally consider antigens to be the foreign substance, but here the case is bit different. That is what we are trying to understand. Now, these antigens are present on the surface of the RBCs. Okay. No one was having much understanding of that. Okay. But for the first time, they tried to find out in a monkey, commonly known as rhesus monkey. Now, in that monkey, what happens is they analyzed the blood sample of that monkey. They found that there is something, a protein kind of substance which is present on the RBC of this monkey. So, on the surface of the RBC, they found one particular protein substance. Okay. Because it was found in this monkey and that starts with RH, hence they started calling that substance, it was a protein and that is basically an antigen. Okay. So, they started calling it as RH antigen. Okay. That is the only reason why it is known as RH antigen. Okay. Now, you saw very same thing was also observed on the surface of RBCs of majority of the people, human beings. What is that majority count? That accounts for 80%, approximately 80% people, whichever sample they took from humans, among that 80% humans were also having similar antigen on the surface of the RBC. Now, that is what we are here to understand. Now, then they later understood if that antigen is available, okay, that means because it is found in the rhesus monkey. So, whichever antigen they have, okay, that antigen, they are calling it as RH antigen. If it is present, then the person's blood group is said to be so and so positive, let us say A blood group, and on the surface of the RBCs of the blood group A, if it is having this antigen, then we call that blood group as A positive. Similarly, if those whom the antigen is absent, then we call that as RH negative, like this. Now, it is because of this presence and absence of the RH antigen that made it to be negative or positive of the respective blood group. Okay. So, that is what we understood. Now, we understood firstly in the first discussion we just understood about blood groups in general. Okay. It is A, B, AB, O. Okay. In the second discussion we understood there is further differentiation or differences in the blood group, that is being a positive blood group or being a negative blood group, that is how happens. So, we understood. Now, we are going to use those two concepts in order to understand this particular concept of blood transfusion, right. So, that is what we are going to do now. So, here there are very specific ways in which blood types must be matched for the safe transfusion. So, what this means is, whenever we are undergoing any blood transfusion from our body to some other's person or from some other's body to our body, let us say, if we have to get blood or if we have to give blood to someone, it has to be done very specifically, that is what it says. Okay. Because it entirely depends on the type of the blood what you have and the type of the blood what the other person is having, right. It is quite simple. Now, then only you can achieve the transfusion to be safe. Okay. So, the right blood transfusion can mean the difference between life and death, that is quite important thing, although it looks very simple, but that is what the right blood transfusion must happen. And of course, before transfusion the blood has to be tested for several infections if any. For example, if there is a person named X and the person needed blood, that is O positive, let us say, and there was another person with the very same blood group, let us say. Okay. Now, you may find it that okay, there is another person with the same blood group, so we can go for blood transfusion. Absolutely, I have no problem, but if the person Y is having some infection in the blood, let us say, if there is some infection in the same person, then the blood transfusion cannot happen. Actually it should not be done, because if the person is having some infection, very same infection can enter this person, right? So, that is also checked much before the blood transfusion. So, there are certain criteria. It is not only just to match the groups, but also to look after the infections if there are any. So, there are several criteria are to be followed. Okay, fine. So, let us see what else we have. So, this is something important again. So, you see, so if the donor is A, that is blood group A is wanting to donate, the recipients can only be A and AB. No one else can take it up. Okay. Now, you see if the donor is B, only B and the AB persons can receive it. That means, the individuals who are having B blood group or the individual with AB blood group can receive blood from the person with B blood group. So, same stands here again with AB. AB is a blood group, only the recipient is AB, same guy. When it comes to O blood group, you see O blood group guy can donate blood to individual with blood group O, blood group A, blood group B and blood group AB. Understood? So, that is the possibility what we have, right. So, I hope so far you understood the three concepts. So, first one was the ABO blood system, second is the presence or absence of the antigen on the surface of the blood groups, third one is blood transfusion. So, we still have a bit more in the very same topic, that is blood transfusion. So, let us have a look at it. So, Rh negative blood, now this is the picture what you have to understand in detail. Rh negative blood is given to Rh negative patients. So, that is the first condition. That means, X person, okay, having let us say some blood group with negative, that means there is no antigen with them, right, that is given with to a person with again no antigen. So, that is possible, that is the first condition. In the very same statement, there is one more condition given here, observe carefully. Rh positive or Rh negative blood. So, it says that second condition says that there is a person with Rh positive, there is one more person, let us say Rh negative. That means, there are two different people, okay, X naught and X1. X naught person, X naught is this is actually the name of the person, X, XY, X naught and X1 or X0 or X1 is the name of the person. Okay. So, when you see X naught person is having positive blood group, some blood group with antigen. Here X1, the person X1 is having some blood group, okay, without antigen. Okay, that is in common I am telling you. So, you see what is given here. So, the Rh positive or Rh negative blood may be given to Rh positive patients. That means, they can give blood to Rh positive patients. So, again I am writing here, you see. So, what it exactly means is, so this statement says that it is only about the blood transfusion, which means it is only speaking about the transfer of only RBCs. In other words, only blood, it is not speaking about the plasma. That is why you see the rules of or for plasma are reverse. So, we will look into those rules later. So, what it says is, so the person named X and the person named Y, right. So, blood is given from X to Y, X naught to Y naught, X1 to Y1. Okay. So, the here the first condition says that a person named X is having some blood group with negative, and that means there is no antigen on the RBC surface, that can be given to the person Y with the same blood group, that is negative. Now, it says here the second condition, a person with, the person name is X naught, okay, and here the person name is X1, let us say. So, if the person is having X naught with a positive antigen, that means in this condition the person is having antigen on the surface of the RBC, that can be given to the person with positive. That means Y naught person can take blood from X naught person, or in other words X0 person with a positive blood group can give blood to Y0 person with a positive blood group, because they both have antigens. Antigens on RBCs. So, remember RBC have the antigen on their surface in the individuals with the blood group positive. The second condition, X1 person can give blood to Y1 person, if the X1 person is having negative blood group, and still it can be given to a positive person, that is what it means. Now, this is all about the blood, right. What exactly blood means? Blood cells, okay. But this game what we have learned, okay, is entirely different when it comes to plasma. So, that is the blood plasma, the rules in the case of blood plasma is reverse, exactly opposite of it when it comes to plasma. So, children, these days you have heard of plasma therapy, right. The plasma of a person infected with COVID, okay, a person infected with COVID, and the person has recovered, and from that the person can take, the doctors can take the plasma, and that plasma can be used for treating another person with COVID, isn't it. So, that is what we have learned, plasma therapy, okay. It is quite successful in the initial days, it was very well, we have seen it was very highly successful. So, that is why we in the plasma of the blood we see antibodies are present. So, antibodies will be acting against the infection, that is why plasma. So, that is why the case with the plasma is entirely reverse, it is entirely opposite of this. So, hope you understood this one. Let us move forward. So, the universal plasma donor has type blood AB blood group, okay, and the universal red cell donor, that is blood we are thinking about, okay, has type O negative blood. That means universal red cell donor, that is only about the blood cells. If you think of, type O is the one who can donate blood, O negative blood can be donated to everyone. Whereas, if it is all about plasma, someone has to donate plasma, if it is AB blood, and that is that plasma we are taking from the AB blood, that can be donated to everyone, that is what it means, okay. So, now you see universal blood donor. So, that was a different concept about, we are trying to understand, in the previous slide we were trying to understand what exactly happens the blood transfusion and what exactly is the difference between plasma transfusion, right. Now, you are going to see universal blood donors, are those with a O negative blood type. Now, I hope it is very clear. Universal blood donors are O negative people. So, the people who are having O negative blood group, they can donate blood to everyone, okay. Now, O negative blood group can be used in transfusion for any blood type, that is what it says. Okay. So, type O is usually in short supply and in high demand by hospitals, because it is one such blood group that can be taken by most of us, is not it? Everyone can take it, especially it has to be O negative. So, that is one reason why it is like that. Okay. So, what is happening here? It is a most common blood type, and at the same time O negative blood is the universal blood type needed for emergency transfusions. Because if a person is having some emergency, some medical emergency is there, okay, met with an accident or something has happened where the medical condition needs the requirement of the blood. In that way, the person can be given with O negative, because if sufficient blood is not given, the person may die. So, that is what is medical emergencies referred here as. So, emergency transfusions, that is one such important need. Another important need, why a person needs O negative blood group, because there are some cases where infants, babies, when they take birth, okay, they will be having immune deficient or deficiency in the immunity. That means, sometimes what happens, babies, they are born due to some reason they do not have sufficient immunity to act against some of the antigens, mixing up from the mother's blood to the baby's blood. So, if these two bloods are different, let us say, okay, then they may not have that ability to fight against that antigens present in the mother's blood. In that case, we need O negative blood group. So, that is one good reason why we need to have this O negative blood group. Okay. So, I want to show you something else about rare blood groups. Now, there are antigens on the RBCs, we learned, is not it? Now, we have known there are more than 600 different types of antigens present on the RBC surface, that is present on the blood cell surface of different individuals throughout the world. Okay. So, when you look here, this presence and absence which creates the rare blood type. So, if there are people around ourselves, we all are having O positive blood groups, okay, and if all around you are having O positive blood group, guys, certainly they would have same type of antigen. So, we are not calling ourselves as rare, but there are certain conditions we have to look into that. So, your blood group type is considered very rare if you lack antigens. That means, you do not have antigens on the surface of your respective RBCs, okay, that 99% of the people are having. It is very simple. Math here is very simple. Your blood group is considered to be very rare only when, for example, you are there in the 100 people, okay, there are 100 of the individuals, okay, I should not use that. Okay. So, there are 100 individuals, let us say, just to make you understand, children, by the way. Okay. So, you see there are 100 individuals, okay. Out of 100 individuals, 99 people, okay, 99 people among those 100 are having positive, that means they have that antigen on their RBC, okay. But there is one individual, okay, the person is not having that antigen. So, negative antigen, in the sense no antigen, then that one person's blood group can be considered to be rare. Hope you understood this time, right. So, your blood group type is considered to be rare if you lack antigens that 99% of the people are positive for, that is only for that particular antigen. It is very simple. What happens, or how do we consider that a individual's blood group is extremely rare? That is what you see here. So, just a math changes here, nothing so difficult. So, if you somehow lack an antigen, you do not have one particular antigen in your blood group, okay, that is present in 99.9% of the individuals around you. 99.99% of individuals around you are having that antigen, okay, they are having that particular antigen, but you are the one, if you do not have that antigen in your RBCs, then you are considered to be, that is your blood group type will be considered to be extremely rare. Okay. So, this is just to make you understand, like it is all about the presence or absence of the antigens. And when we say antigens, you can see here there are different types of antigens, right. So, it is not only one or two type, they are, basically antigen is more or less like a structure, that is like a protein. If it is present there, you will get to see that that is the type of antigen what you have, right. Based on that, your blood group will be decided whether it is a regular blood group or it is a rare one or it is extremely rare, right. So, if you see here, now I am going to show you the blood group type and the population. So, this is exclusively with respect to the Asian population, okay, Asia. So, if you look at the population with A positive blood group, that accounts for 27% of the individuals, and when it comes to B positive, 25% of the people are having B positive blood group. When it comes to AB positive, that accounts for 7%, and when it accounts for O positive blood group, that accounts for 39%. Okay. So, this is all about positive, that means those blood groups which have antigen on their respective RBCs. When it comes to blood group with negative, that means no antigens. So, you remember this, lack, you lack antigen, again here somehow you lack one particular antigen. So, there is no antigen on your surface of RBC, that is what is making you very rare. So, A negative, 0.5% people, in the Asian context, they do not have antigen in them. So, B negative people 0.4%, AB negative accounts for 0.1%, O negative you see accounts for only 1%. So, that is why you get to see these blood groups are rare. That is why we have blood banks and all to store them and to use accordingly. Okay. Now, we have to see the process of blood coagulation. So, a clot or coagulum is formed mainly of network of threads. The name of those threads are fibrins. Now, it is not just that simply it forms fibrins, there is a process for that. Okay. So, whenever a blood vessel or you get scratch or wound while you are playing, that is when you get to see bleeding, and that has to be stopped. To check the blood flow which is unauthorized, we have a system called blood coagulation system. So, firstly, a prothrombin is a protein, it is more or less like a protein, it has to be get activated. So, it has to form thrombin, that can only takes place in a reaction with an enzyme as a catalyst, that is thrombokinase. Thrombokinase is a catalyst. Then you see here fibrinogen is another protein present in the blood plasma. It has to convert itself into fibrin. Okay. If it has to convert itself into fibrin, we have to have thrombin as the enzyme which catalyzes the reaction. Now, it is this fibrin mainly involved in the clotting of blood. As you can see here, fibrins. So, this is in short to make you understand that. So, not only these reactions, but also you need to have the calcium ions, they take equal part in blood coagulation process. So, I want to show you that. So, you see this is a normal blood vessel. So, when you see this normal blood vessel having no rupture, it is a proper tube. Okay. So, when you see this particular blood vessel, it is actually broken. So, you see blood cells will be flowing out. This flow of blood cell is unauthorized. Certainly, that should not happen. We are losing blood, we are losing components of the blood as well as pressure built in the blood. So, we need a system to clot the flow of the blood, that is what is blood clot, and you have seen how the process happens which leads to the formation of fibrin. So, these yellow structures here you can see that leads to the formation of the mesh like arrangement to stop the flow of blood, right, that is what happens. So, that is why you see here it is a repaired vessel wall. So, in this way to stop the flow of the blood, that is how it happens. So, we have some more topic here. So, this is what is the circulatory system we have in humans. Okay. So, we get to see that in humans, and you see there are two different loops. Okay. Two different loops are available. So, either way you see there is a common point, that is heart. So, from the heart one loop goes here. Okay. This is actually known as pulmonary circuit. So, this loop 1, loop 2. Loop 1 is pulmonary circuit. Okay. Loop 2 is systemic circuit. Okay. So, these are the two different types of circuits, blood circuits we have in our body. We will be discussing that in the upcoming session. Okay. And I would like to show you the other types of circulatory system in different organisms. So, when you see here, so when it comes to the circulatory system in fishes, because they have two chambered heart, they have single circulation. So, that means they do not have two loops, they have only one loop. In our case, we have two loops. So, we will have double circulation. So, when you understand here, we have amphibians with three chambered heart. So, incomplete double circulation. Reptiles again they have three chambered heart. Usually they have incomplete circulation except crocodile which has four chambered heart. Following the birds and mammals, birds and we have the same way, that is double circulation. So, this is in the presence of the chamber count, you see the types of circulation, and when you see in different organisms there are different types of circulatory systems, starting with open circulatory system and the second one is closed circulatory system. So, in this type of blood flow you will see that there are arteries. You get to see in the open blood flow happens from the heart into the arteries, that is a common thing. But the difference here is these arteries would open into a large space called as sinuses, that is a open space known as sinuses, and the very same sinuses there is no additional network between the arteries and veins. That means you do not have capillaries available here. That means that is what, no interconnecting vessels. So, this kind of system we get to see, that is open circulatory system in arthropods, mollusks and echinoderms. Arthropods accounts for insects, mollusca is for snail, octopus, squid, those kind of organisms. Echinoderms are for starfishes, sea cucumber, or you get to see the the sea urchins. When it comes to closed circulatory system, we have a network like that, where arteries, veins, they have separate tubes. Along with that, they have interconnected network known as blood vessels, capillaries. So, that is what the system we have. So, our system is a closed circulatory system. So, we get to see in annelids and chordates. So, we belong to chordates, right. So, hope you understood this. So, let us look into the topics what we have dealt so far. So, in this particular session, we looked at the discovery of the blood grouping, following which we understood about the discovery of the antigen and how it affects the blood groups, and we looked at the blood transfusion and few details about the universal donor, following which we saw different blood groups over a population, that is Asian population. So, that is what we understood, and we understood the process of blood coagulation, how actually blood clot occurs. And then we saw various circulatory systems and the type of circulatory systems what we and other animals have, right. So, hope you understood this session. So, we will be discussing more and more topics in the upcoming session. Thank you.